All right, so today we're going to learn how to do a DiHybrid cross. So first, let's start by dissecting the title DiHybrid. So di means two, and hybrid is just the offspring of two different parents. So this time we're going to look at the combination of inheriting two traits from parents. So this could be like color and height. Okay, so is it going to be like a green tall plant or a green short plant? Or yellow tall or yellow short? So this time, when we look at DiHybrid crosses, there's going to be 4 types of allele from each parent. And so it's 2 traits and then 2 alleles per trait. So 4 total. So the example we're going to look at today is still going to be using pea plants, but this time we're going to look at the color and the shape of pea plants. So the dominant color is yellow, which will be assigned a capital Y. The recessive color is green, which is a lowercase y. The dominant shape is round, which is assigned a capital R. And the recessive shape is wrinkled, which is a lowercase r. So for our first cross, we're going to cross a homozygous dominant by a homozygous recessive. So the first thing I'm going to do is draw a Punnett square. And this time, since it's a dihybrid, we're going to make 16 boxes. So I just start by dividing my box in half. And then I divide it in half again on each side. So I have 4 columns. And now I'm going to make 4 rows. So by the time you've set up your Punnett square, you should have 16 boxes inside. Now, the next step is going to be to figure out, well, what are the actual alleles for homozygous dominant by homozygous recessive? Okay. So I'm going to use the traits that were listed on the previous slide. Okay. So homozygous means the same. I'm going to start with color just because color was listed first. So homozygous dominant. So the dominant color is yellow. And it's homozygous, so it has to be 2 capital Y's. And then for shape, the dominant trait is round, which is a capital R, and homozygous, so 2 capital R's. And then the other parent is homozygous recessive. So the recessive color is green, which is lowercase y. And then the recessive shape is wrinkled, which is lowercase r, lowercase r. So now, I know it might be tempting to just copy these 4 letters and put 1 in each column. However, that's not how it works for the DiHybrid cross. You have to have 2 alleles per column or per row. So in order to figure out what goes in each column or row, we're going to look at our formulas right here. And so what I'm going to do is the color is going to combine with the shape. So I'm going to get capital Y, capital R. Then this same Y is going to combine with the second R. So capital Y, capital R. Then my second Y combines with the first R, still capital Y, capital R. And my second Y combines with the second R. So capital Y, capital R. Now I'm going to do the same pattern for the other side. Okay, so this time it'll be the first Y combines with the first R, so little y, little r. The first Y combines with the second R, so little y, little r. The second Y combines with the first R, so little y, little r. And then the second Y combines with the second R, so little y, little r. All right, so now I'm going to go ahead and cross these alleles just like I did in the monohybrid. So I'm going to start in this square. And this square is just the combination of these alleles in this column and these in this row. So it's going to become capital Y, lowercase y, capital R, lowercase r. And I'm just doing my colors first, my Y's first, my shapes second, just how they've been the whole time. And then I'm trying to write the capital letter first for each. So now I'm going to do this box. It'll be the combination of these set of alleles and this set of alleles. So I'm going to get capital Y, lowercase y, capital R, lowercase r. This box, capital Y, lowercase y, capital R, lowercase r. Capital Y, lowercase y, capital R, lowercase r. Now I'm going to start the second row. Capital Y, lowercase y, capital R, lowercase r. If you're starting to see a trend, they're all going to end up being the same because the alleles in each square are the same. So every single one of these is going to be a capital Y, a lowercase y, a capital R, a lowercase r. Okay, so I finished the Punnett square. So every single box says capital Y, lowercase y, capital R, lowercase r. So the genotype for all offspring is capital Y, lowercase y, capital R, lowercase r. And then for the phenotype, I'm going to look as the capital Y is color, which is yellow. And then the capital R codes for round. So every single one of those seeds is going to be yellow and round. All right, so now let's continue with our example. This time, let's take two homozygous seeds and let's cross them. So I'm going to draw my Punnett square. And I'm going to go ahead and divide it into the 4 columns in the 4 rows to give me 16 boxes. And then now, remember, before you just start placing letters, you have to combine the alleles here to figure out what goes over the rows or the columns. So to do that, big Y is going to combine with the big R. So Y, R. Then the big Y will combine with the little r. So big Y, little r. The little y with the big R. So little y, big R. And then little y, little r. And now I'm going to do the exact same thing for the rows. So big Y with big R. Big Y with little r. Little y with big R, and little y with little r. All right, so now that I have my columns and my rows labeled, I can go ahead and I can start filling in the genotypes of my offspring. So I'm going to start in this box, and I'm going to combine my Y's and then my R's. All right, so this one I'm going to have capital Y, capital R, capital R. The next box should be capital Y, capital R, lowercase r. Then it's capital Y, lowercase y, capital R, capital R. Capital Y, lowercase y, capital R, lowercase r. Second row, capital Y, capital R, lowercase r. Capital Y, lowercase r, lowercase r. Capital Y, lowercase y, capital R, lowercase r, capital Y, lowercase y, lowercase r, lowercase r, capital Y, lowercase r, capital R, capital R, capital Y, lowercase y, capital R, lowercase r, lowercase y, capital R, lowercase y, uppercase R, lowercase r, capital Y, lowercase y, capital R, lowercase r, capital Y, lowercase y, lowercase r, lowercase r, lowercase y, capital R, lowercase r, lowercase y, lowercase r, lowercase r. All right, so this one has a lot of variation. So now we're going to look at the genotypes and the phenotypes present in this whole table. I need extra space, so I'm going to go ahead and change my slide to record the genotypes and the phenotypes. All right, so now we're going to look at the phenotypic ratios of the offspring that we get when we cross two heterozygous parents. So in order to answer this question, we're going to use the Punnett square that we just filled out on the previous slide. So if you don't have that written down yet, go ahead and rewrite or rewind this video and copy that Punnett square down. So there are a lot of different genotype options, so we're just going to focus on phenotype. Okay, so our phenotype options are for a yellow round seed, a green round seed, a yellow wrinkled, and a green wrinkled. So the genotype to support yellow round, you have to have a capital Y for a yellow and then a capital R for round. This line here, it could either be a capital or lowercase y, and this second line would be a capital or lowercase r. The next phenotype is green and round. So for genotypes, in order for it to be green, you need 2 lowercase r's. And then for round, you need at least 1 capital R. And then the second letter, it could either be a capital R or a lowercase r. Our third phenotype is yellow wrinkled. So for yellow, you have to have at least 1 capital Y. The second letter could be either a capital or a lowercase y. And then for wrinkled, you need 2 lowercase r's. And then the fourth phenotype is green and wrinkled. And so for green, you need lowercase y. And for wrinkled, you need lowercase r. So now to figure out the ratio, we need to look at the genotypes that we wrote down in the Punnett square on the previous slide. So now I'm going to go back to the first one, the yellow round, and I'm going to count how many genotypes start with a capital Y and have either a capital or a lowercase y, and then a capital R and either a capital R or a lowercase r second. So when I look at the Punnett square, I count 9 different genotypes, and the total number is 16. So our ratio is 9 to 16. Next is green and round. So I'm going to look at how many genotypes are YY capital R, and then either capital R or lowercase r. So when I look at the Punnett square, it's a 3 to 16 ratio. Next, I'm going to look for yellow wrinkled, which is a capital Y and either a capital or lowercase y, and then 2 lowercase r's. So when I count those squares, there's 3 of them out of 16. And then finally, I'm going to look for the genotype lowercase y, lowercase r, and there is only 1 box with that genotype, so it's a 1 to 16 ratio. And so we can simplify this as a 9 to 3 to 3 to 1 ratio.